Um, Hello guys, today we are going to continue our revision for final exam and to stop till chapter 9. We will start with section 1, which is manipulating DNA. As we learned that the biotechnology relies on cutting DNA at its bases, and the scientists use several techniques to manipulate the DNA because DNA is very large molecule, but it's still a molecule. It is far too small to see, so they can not pick it up with you cannot pick it up with your hand. So the scientists use can work with the DNA without being able to see them or handle them by using chemicals, by using computers, and the bacteria is a very important part to manipulate the DNA. We said that the chromosome can be sliced into pieces, but it cannot be sliced by using a scissor. Instead, they use enzymes to cut the DNA, and these enzymes are called restriction enzymes. These enzymes come from various types of bacteria. This restriction enzymes allow the scientists to more easily study and manipulate the genes, and it cuts the nucleotide sequences by restriction enzymes at restriction sites. As you see here, we have different restriction enzymes cut DNA in different ways. Each enzyme has a different location on the DNA to cut on. We call it restriction sites. Some cut straight across the leaf, blunt ends, and some make sticky ends like this one. Okay, uh, this one will be more useful because it can bind to the other complementary nucleotide. Now here, restriction marks show the length of the DNA fragments. And after cutting the DNA to different segments, there are restriction marks that can be used to show the length of the DNA fragments by using the gel electrophoresis. To separate, first we have to separate the DNA fragments. The DNA sample is cut with restriction enzyme. Then electrophoresis is used by using an electric current which pulls DNA fragments through a gel. Okay, the length of the DNA fragments can be estimated from the distance it travels. Whenever the fragment is smaller, that means it can travel more distance. Whenever the fragment is larger, that means it travels less distance. As you can see here, this is the electrophoresis. Smaller fragments move faster and travel farther than the larger fragments, and different fragments appears as bands on the gel as you see here. Now you can see here different DNA fragments with this different sizes. Now restriction map is very important because it indicates the size of the sequence, but not the DNA sequence. It is useful in genetic engineering and it is used to study mutations. Why? Because the mutations may add or delete bases. Okay, بالفريم shift مثلا. So between restriction sites, there is if this something happens, it changes the length of the DNA fragment, and you can predict that there is a mutation, okay, in the DNA. Now let's go to the section 2 in chapter 9, and section 2 is talking about copying DNA. We learned that DNA replication happens normally in each cell, okay? But there is a scientist who found a technique that can help in copying DNA outside of the body, okay? And by using a polymerase اللي هو DNA polymerase by a procedure or a technique called polymerase chain reaction. This technique is used to to make many copies of the DNA segments to make it more easier for the DNA segments to be studied. Now, forensic scientists use the DNA from cells in a single sample, like hair in a crime scene, to identify a criminal. The scientists compare the DNA from different species to determine how closely the species are related. Okay, so The original amount of DNA from any of these sources is far too small to accurately study. So samples of DNA must be increased by using this technique اللي هو the PCR polymerase chain reaction to amplify the sample and to make it more accurately studied and analyzed. PCR polymerase chain reaction uses polymerases to copy DNA segments. PCR makes many copies of specific DNA sequence. It makes copies of a certain gene. If you need a certain gene, it can copy this gene for you. Okay, it amplifies DNA samples. PCR is similar to DNA replication. As in the COVID-19, they can detect the gene of this virus by copying this sequence of DNA using the PCR. They amplify the sample to make sure what the amount of the virus inside and if it's inside your body or not. Now PCR has 3 steps. It uses 4 materials. أول شيء it uses the DNA to be copied and DNA polymerase and nucleotides which are adenine, thymine, cytosine, guanine and 2 primers. The 2 primers are important to let the DNA polymerase to start copying the sequence. The 3 steps of PCR occur in a cycle. The first one is separation, in which heat is used to separate double-stranded DNA molecule, then binding the primers to each DNA strand in opposite ends, and then copying the DNA by DNA polymerase, which binds the nucleotides together to form new strand of DNA. Each PCR cycle doubles the number of DNA molecules. All right, so we come to the next section, section 3, DNA fingerprinting. Now, DNA fingerprinting okay, just a minute, guys. All right. DNA fingerprinting identifies people at a molecular level. We know that a couple of decades, the lines and the swirls of someone's fingerprints were detected, best hope for identifying a person. But now the investigators gather biological samples and analyze DNA for another kind of evidence. So DNA fingerprinting, which is a representation of part of individual DNA that can be used to identify a person at a molecular level. A DNA fingerprint is a type of restriction map also, but based on parts of individual DNA that can be used for identification. It's based on non-coding regions of DNA, not the coding, non-coding. This is very important. Non-coding regions have repeating DNA sequences. The number of repeats differs between people. Okay, as you see here, this is a DNA sequence. Maybe this DNA sequence have different repeats from one person to another, as you see here, and it happens or it can be used or based on non-coding regions of DNA. It becomes a reliable and widely used process because the specific nucleotide sequences that are repeated can be found in everyone. But more importantly, from one person to another, the number of repeat sequences can differ greatly. Okay? DNA fingerprinting compares at least 5 regions of the genome. Yani you should compare 5 regions at least. In that way, it's more certain that the patterns of DNA fragments in the fingerprinting is unique. And sometimes انت بتلاقوا في عندي the same repeats at some segments. To make sure لازم نعمل comparing at least 5 regions of the genome or the DNA segment. DNA fingerprinting depends on the probability of match. Many people have the same number of repeats in a certain region of DNA. But the probability that 2 people share identical numbers of repeats in several locations is very small. عشان هيك I have to choose 5 locations at least to compare between them. Okay, now let's go to section 4, genetic engineering. Genetic engineering that means the DNA sequence of organisms can be changed. Sometimes they change in the genes or the sequence on the DNA to make better traits for organisms. And the first idea comes from cloning. Cloning occurs in nature through asexual production. And what the meaning of cloning? Clone is genetically identical copy of gene or of an organism. Cloning cannot happen naturally in mammals. Okay, it can be cloned by a process called a nuclear transfer by taking a nucleus or removing a nucleus from an egg, and the nucleus of a cell from the animal to be cloned is implanted inside, and then the unfertilized egg is taken. First, the egg is taken. He an unfertilized egg from an animal. The nucleus removed, then the nucleus of the cell from the animal to be cloned is implanted. But then, the egg is stimulated. If the procedure is successful, the egg will begin dividing, and after the embryo grows for a few days, it is transplanted into a female. From this idea, the geneticists can make new genes to be added in organisms. Genetic engineering involves changing an organism's DNA to give it a new trait. Genetic engineering based on the use of recombinant DNA. Recombinant DNA هو contains genes from more than one organism. يعني عشان أعمل أنا change بالجين أو بالدي ان اي of an organism لازم يكون عندي recombinant DNA. يعني الدي ان اي في الدي ان اي الأصلي وفي مع genes from more than one organism. Okay? هلا البكتيريا البلازميد are often used to make recombinant DNA. في عندي plasmids are a closed loops of DNA that's separated from the bacterial chromosomes. They can replicate on their own within the cell. بياخدوا restriction enzymes cut the plasmid and foreign DNA gene inserted into the plasmid. بدخلوا الجين اللي بدهم اياه يضيفوه على هذا البلازميد. تمام؟ as you see here. هلا هاي البكتيريا اذا اخذنا البلازميد تبعها وحطينا و inserted a new region او جين عليها معناته صار هذا اسمه transgenic organism. لانه هاي البكتيريا صار عندها one or more genes, يعني more than one gene from another organism inserted to its genome. And how we make this for bacteria, we can make this for plants and animals. Referring to the previous figure, we observed that the plasmid and the foreign DNA with the gene are cut with the same restriction enzymes. بعدين هدول sticky ends of the plasmid and the foreign طبعا البلازميد هي DNA والجين اللي أنا حطيته هو عبارة عن nucleotides برضه part of the DNA. They will match together the sticky ends. The plasmid and the foreign gene are bonded together to form recombinant DNA. هلا transgenic bacteria can be used to produce human protein. Okay? كيف؟ gene inserted into plasmid, the plasmid inserted into bacteria. هلا in a way, bacteria are turned in tiny gene factories that make copy after copy of the plasmid. As a result, the transformed bacteria make many copies of the new gene. بتبلش البكتيريا تعمل many copies of the new gene and express the gene. A transgenic organism has one or more genes from another organism inserted into its genome. مثلا الجين of the human insulin can be put in the plasmid. بياخدوا plasmid تاع البكتيريا and they add this gene تبع الهيومن انسولين على البلازميد تاع البكتيريا. هذه اللي بتصير عندي البلازميد تاع البكتيريا صار دخلناه جوا البكتيريا صار عند البكتيريا ترانسجينيك بكتيريا لانها عندها جين من الهيومن. هاي البكتيريا when it have this new plasmid or recombinant DNA مع النيو gene, it can make a human insulin that is controlled, collected and used to treat people with diabetes. إذا كمان مرة هذا is very important in making insulin. بياخدوا الجين الهيومن بحطوا هذا الجين تاع الهيومن على البلازميد تاع البكتيريا and insert it again to the bacteria. هلا البكتيريا صار عندها new gene to be expressed اللي هو making insulin. بتصير البكتيريا expressing this gene and making more insulin. Then they can collect this insulin from the bacteria يبيعوه الانسولين اللي بيستعملوه مرضى السكري as a medicine. And a transgenic plant change a plant's DNA نفس الشيء الجين اللي بدي انا اغيره في البلانت، الجين is inserted into plasmid، والبلازمid inserted into bacteria، بعدين بيخلوا البكتيريا to infect the plant، والنيو gene will become part of the plant DNA and express like another gene. هلا transgenic plant ممكن يكون ايش هدفها؟ increase the food production to save endangered species. مثلا بيكون في عندي جين اسمه البي تي توكسين، هذا بحطوه في البلازما بتاع البكتيريا and this is important، بيعمل كانه pesticide، ما بيخلي الانسكتس to infect the plant. And it is uncommon in agriculture. But transgenic bacteria infect the plant expresses foreign gene, and many crops are now genetically modified. Transgenic animals are much harder to produce than GM plants. GM means genetically modified، يعني صار عليها تغيير في الجين because animals are more resistant to genetic manipulation. To produce transgenic animals, they must first get a fertilized egg cell, then the foreign DNA is inserted into the nucleus, and the egg is implanted back into the female. فأصعب الطريقة هاي. Examples of transgenic animals زي مثلا بيعملوا عندي تغيير في الجينات في some cows to yield more milk, high yield for milk producing cows, to produce milk with similar properties as a human breast milk. Or they developed a genetically modified dairy cattle to grow without horns. ممكن يعملوا عندي مثلا transgenic animals to make fast grow salmon. Transgenic animals also used to study diseases. They can use onca mouse to study cancer and anti-cancer drugs, as we said. But at the same time, scientists have concerns about the uses of genetic engineering. Can be some concerns they should keep in their mind, like the possible long-term health effects of eating GM food. And if we keep eating genetically modified, not naturally food, they are genetically modified food, this may have possible long-term health. We don't know. They have to keep in concern. And possible effect of GM plants, genetically modified plants, on ecosystem and biodiversity. Now let's go to section 5. In section 5, we will come to genomics and bioinformatics. We said that entire genome are sequenced, studied, and compared. Genomics involves the study of genes, gene functions, and entire genome. قلنا الجينوم هو the whole amount of the DNA, and genomics هو the study of the genome. Can include the sequence of genome. Genomics study of genome means and you can study the sequence of the genome and compare genomes within and across the species. Gene sequences is determining the order of a DNA nucleotides in genes or genomes. احنا قلنا إنه all the living things إنهم the same DNA structure. What is different among these organisms هو sequencing of the nucleotides. Okay? فأنا عندي الجينوم وعدد الجينز كمان. الجينوم اللي هو most DNA. Okay? most DNA amount هي الجينوم. هلا الجينوم of several different organisms have been sequenced. فطلع عندي different total DNA for each species, as you see here. They are all made of the same nucleotides but with different amount and different sequences. هلا كان في عندي كمان human genome project that sequenced all of the DNA base pairs of the human chromosomes and they analyzed DNA from a few people, and they're still working to identify and map human genes. Technology allows the study and comparison of both genes and proteins. Bioinformatics is the use of computer database to organize and analyze biological data. Okay, how this happens? After having the sample of the blood, you have to extract the DNA, and the DNA has sequenced by a sequencing machine, and this is connected to a computer database. Having a computer database will organize and analyze the biological data. So it's easy for you to, or for the technician to compare between the samples and to see the whole sequence of the DNA. There is also DNA microarray that used to study the expression of many genes at once. We would like to check, which have lots amount of information. وبروتيوميكس هم study and comparison of proteins اللي هم sequencing of amino acids. Okay, the last part included in our exam is section 7, section 6, which is genetic screening and gene therapy. We said that genetics provide a basis for new medical treatment. Okay, and anyone could be carrier of genetic disorder. Genetic screening detects genetic disorders. It's important to detect if there is a genetic disorder by genetic screening. And as we learned, it is a process of testing DNA, okay, by different methods to determine person's risk of having or passing disorders. Now, genetic screening involves many methods like pedigree and family histories as well as DNA testing. It can detect genetic disorders, okay? It involves testing of DNA by using electrophoresis. It determines the risk of having or passing genetic disorders used to detect specific genes or proteins. They can detect some genes related to an increased risk of cancer or other disorders like the Duchenne muscular dystrophy. هلا gene therapy is replacement of faulty genes. Now, if we make a genetic screening and we detect any fault in some genes, researchers found a way to treat some genetic disorders by a way called gene therapy. So gene therapy who were replacing, detecting or missing genes, بياخدوا الجينز اللي هو فيه defect أو miss and replace it with a new genes عشان to treat the disease. And this gene therapy cannot be used for all the disorders. Till now, they found gene therapy for certain disorders. How this happens? First, they get the new gene into the correct cell of patient. Once in the body, a gene has to become part of the cell of DNA. And one method, they try to take sample of bone marrow, known as stem cells, and infect them with a virus. هذا الفيروس بيكون genetically engineered with the new gene. Then the stem cells are put back in the patient bone marrow because they can divide and make more blood cells within the gene. As you see here in the figure, هذا الفيروس is called vector. هذا modified, genetically modified, يعني هو transgenic virus لأنه الفيروس أصلا صار في عنده they add some genes to its DNA فصار modified. They take this virus, they add to the virus a gene that they wanted, then this virus will infect the cells of the human, like the stem cells of the bone marrow, and this will start to express the gene inside the body or inside the cell. وهدول cells all, when they divide, will have this gene which is expressed. And in this way, this gene or this defect of the gene will be treated. The first successful trial of gene therapy took place in 1990. The treatment was used on 2 children with genetic autoimmune disorder, and the children are now adults leading normal lives. Several experimental techniques are used for gene therapy. زي عندي يعني بيستعملوا جين ثيرابي أكثر من تكنيك. أول شيء ممكن يستعملوا genetically engineered viruses. هدول الفيروسز بيغيروا بالجينات تاعونهم. Then they will infect يعني they add the gene they wanted للفيروس DNA. يصير هذا الفيروس اسمه genetically engineered يعني يتغير عندي الجين تبعه في بالجينات صار عنده بالDNA جين from other organisms. هذا genetically engineered virus will use to infect a patient cell. هلا برضه في another technique insert gene to stimulate immune system to attack cancer cells. وفي عندي insert suicide genes into cancer cells that activate a drug so that only cancer cells are killed. Okay, guys, so we stop here. I wish you good luck, all of you. Study well and We'll see you later. Take care.